Thank you very much for having me here. I appreciate the opportunity to come and meet with a lot of your farmers. APRICID has been hosting me for several days now and interacting with a lot of different folks. And I want to thank them most of all, and I've really been enjoying my stay. I'll be talking a lot about the cover crop research work that we do in the U.S. I'll try to talk slowly. I know you have an interpreter, and I talk very fast. Oh. Oh, okay, gotcha. All right, great. So there we go. Great, so I'll be talking a lot about the role of cover crops in nitrogen management and the challenges in fitting them into a rotation and the benefits that we get from cover crops. Okay, all right. So it did not come up in English. But, okay. I can try. Yeah, yeah. Okay, so no English for me, only Spanish. I'll try to remember. I give this presentation a lot, so I remember most of my slides. Some of them I won't remember every bullet and every detail, so I will try. But what are we trying to do with cover crop-based cropping systems? We're trying to maximize productivity and profitability. That's the most important. If we can't maximize productivity and profitability, farmers are not going to adopt cover crops. But there's other benefits that we get from cover crops. They help with suppressing weeds. They help with providing nutrients. They can increase water infiltration and storage. And they can also make cropping systems more resilient to climate change, particularly as we have more droughts and floods. Cover crops can play an important role in mitigating against that. Still learning here. Okay. So there are a lot of benefits to cover crops. There are short-term benefits, and there are long-term benefits. When we think about the short-term benefits and trying to get farmers to adopt cover crops, we want them to be immediately profitable, right? If they're not immediately profitable, there's not as much interest in using them because a lot of farmers are leasing their land, so they don't know if they're going to be on the land the next year. Investing in that land is not their top priority. So things like forage, if they can get an immediately benefit, that makes a big difference. Getting nitrogen from fixing cover crop, that's going to provide some immediate benefit. In the case of water, we can often get greater water infiltration and storage, so that can provide some immediate benefits, as well as insect pollinators and bringing in beneficials. But as we build our soils, and we're cover cropping for a longer period of time, we start to see more benefits. As we build our organic matter and increase the carbon that's in the soil, then we can start to see benefits where we are building the soil health and we're building our resiliency to climate change so that maybe not every year we see a yield difference, but there is a more frequent benefit with cover cropping than in the long term than not having them in the system from a carbon storage standpoint. And again, as we also build our organic matter, we also get more water holding capacity, and so this for the storage. I keep advancing the wrong direction. All right, there we go. So there are many benefits to cover crops, and this is just a theoretical schematic that suggests that maybe one cover crop can do everything. If these bars go all the way to the end, it makes it seem like one cover crop is the super cover crop and can do it all. But we know that's not the case, right? Not one cover crop can do everything. This is just theoretical that these are the types of services that cover crops provide. And there are many different species of cover crops that we can be targeting to achieve those benefits. When we think in terms of the benefit of cover crops, stay right here? Okay, stay right here. They want me to stay right here. I'm moving too much. All right. So when we think in terms of the benefit of a cover crop, we really need to think in terms of the biomass. Sure, there are a lot of other benefits from cover crops when they're small, and there are some different services, but generally there's a pretty strong relationship between the amount of benefit we get and the amount of biomass that we have out there. And so this is not a quantitative figure. I just made this and I dropped these pictures in this place, but generally when we have a little bit of biomass, we get a little bit of erosion control. As we start to get more biomass, maybe we get some more erosion control and a little bit of better water infiltration. But it's as we start to push the biomass and increase the amount that we have that we start to target, provide more forages, we get more nitrogen, and ideally we can also get some weed suppression benefits the more biomass we have out there. So now I'm going to flip this figure around. Before we were looking at the arrows going out. And when the arrows were going out, that was talking about the benefits that cover crops provide. Now what I want you to do is think about what it takes to get there. And so with cover crops, the way in which we manage them drives everything. If we manage our cover crops as we do when we think about our cash crops, and we focus on the details, because cover crops are a plant, they're just like every other plant that we work with that's a cash crop. It responds to the same environmental conditions, it requires the same things to grow, and the more time and attention to detail, you start to see more benefits. And so here you can see the planting methods, the nutrients, the types of genetics that we use, the cost of the seed. All of these drive the performance of the cover crop. And when we think about performance, I want you to think about quality and quantity, because that pretty much drives everything. What is the quality of the cover crop, and how much do you have? That drives everything that goes on in the landscape with cover crops. And when we think about quality and quantity, I like to think in terms of that there are intrinsic factors that just drive the potential. So your climate and your soil are going to limit your potential. There's not much you can do about that. You can improve your soil some, you can increase the soil health and get some more benefit, but generally your climate and your soil dictate what your potential is. And then management gets you somewhere to that potential. So most farmers are not achieving the maximum potential. Their management is less than optimal, and they're reaching some level below that. And along that scale is where you start to see the differences in the benefits you're trying to achieve. So now we're going to just talk a lot about grasses. We're going to talk about legumes. I'm going to introduce them. Again, I'm sorry. I don't read Spanish very well, so I don't know what my slides are saying, but I'll try to remember most things. But generally, grasses have a fibrous root system. They grow very fast. They're good at nitrogen scavenging, and they're very winter hardy. So that's why they're very widely adopted. They're easier to fit into the rotation, and farmers are willing to work with them because they have much more flexibility. But they don't decompose very fast, and they really don't provide much nitrogen. They're more scavenging nitrogen and retaining them in the system. So this is a table that I put together that's being used in an adaptive decision support tool for recommendations on cover crops in the U.S. And this gives you a sense of the performance potential of a cereal cover crop. You can see we looked at several different cereals to come up with these values. And what I mean by these different low, medium, and high ranges, that generally means your soil fertility. If you have a lot more organic matter, if you have a lot more residual nutrients, particularly if you have animals in your rotation, if you have livestock, you're going to have a lot higher nitrogen in your soil, and the more nutrients you have in the soil, the better the cover crop performs. So this is at the tillering stage, right? So at the tillering stage, the cereal rye is very small. That's when you plant it in the winter, and early spring before it greens up. Well, we get green up. Where I was the last couple of days, it doesn't look like you get much green up. It seems like it grows all through the winter here. But generally, the tillering stage is while it's short, and then stem elongation is as it starts to elongate. It starts to grow up. And then this is when it's flowering here, right? and this is the range of biomass. But what I really want you to focus in on is these carbon to nitrogen ratios. You see that at the tillering, the carbon to nitrogen ratio is very small. It's 18 to 1. That's the amount of carbon you have to nitrogen. So when we have a low carbon to nitrogen ratio, we have a very high quality material. When we have a high carbon to nitrogen ratio, we call that a low quality material. So let's look at this, for example, here. This is a cover crop at tillering, and this is in the boot stage right here. And you can see the differences in the amount of biomass, and you can see the differences in the carbon to nitrogen ratio. They're going to have differences in how much they provide nitrogen or decompose. So generally, we like to think that below about 20 to 25 parts carbon to 1 part nitrogen, you're going to be releasing nitrogen. Whereas if you're above that threshold, the microbes are hungry for nitrogen and they don't have enough. And so the microbes are waiting for more nitrogen. One microbe dies, the next microbe eats their brother, and then he gets that nitrogen, and it just stays in the microbial biomass. And so that nitrogen is not available to the plant. And so we think of this as the mineralization, immobilization transformations, that above about 25 parts to 1, you're immobilizing, you're tying up nitrogen, and below, you're releasing it. So let's look at what the soil profile looks like coming out of a cereal rye cover crop like this. This is in Maryland, where I live. Our soils are not as good as what I've been seeing in Rosario. You got really nice soils with high organic matter content. My soils tend to have lower and are a bit sandier. But you can see here that this is a bare ground, so that's no cover crop. And this is, oops, I think I went too far. Nope, I guess all right, did we just, it got switched. All right, okay, I see. This is, just ignore this. I had a white bar over this, but it didn't come out for some reason. Just ignore this. Just look at these two. This is the bare ground, and this is how much nitrogen is in the profile in the spring, right when we kill this cover crop, this early cover crop. So this is killing that cover crop when it's at the tillering stage. And you can see just the presence of the cover crop reduced the amount of nitrogen in the profile considerably. And that nitrogen is tied up in the cover crop biomass. But if we let that cover crops grow, now what you're looking at is the soil in late spring, early summer. So this is right when we go and plant our cash crop. Here this would be when we're planting maize. And so now you're seeing the difference here is this bare ground has had no cover crop all the way till the end of spring, early summer. This cover crop was killed early, and now you're looking at what the soil looks like 4 to 6 weeks later. And then this is what the soil looks like right when you kill that cover crop at the end of spring, early summer. And you see big differences in the amount of nitrogen that's in the biomass and what's left in the soil. Let's switch to legumes. So as we know, legumes fix nitrogen. They have very low carbon to nitrogen ratios, and they release their nitrogen very quickly when they're terminated. They are not as fast release as mineral fertilizers, but they do release nitrogen very fast. So we have to realize that even though legumes are organic material, not like a mineral fertilizer, they still decompose and often release nutrients fast, but not as fast as a mineral fertilizer. And the benefits, obviously, compared to manure, is that it's not bringing in new phosphorus, and it doesn't have the volatility issues which can affect your nutrient use efficiency on the farm. Now let's look at what those rates look like. So here now we're just looking at the legume, and this is an early kill, an intermediate kill, and this would be about flowering. So this is when the cover crop is full flowering. And these are the types of biomass we get on poor, intermediate, and high-quality soils at these different kill dates. But notice how the CN ratio doesn't really change much. And that's important, that the quality is always very high. So let's look at a study that I did across the Northeast U.S. with a lot of colleagues of mine, and looking at how the hairy vetch seeding rates, planting dates, and termination dates affected its biomass potential. So what I want to do is first start with just one site. This is where I live. And this is, up here you're seeing the latitude differences between the different sites. So you can see we have much colder sites than where we are in Cordoba, and we have warm sites like where we are here in this region. And so what you're looking at right here is an early kill, an intermediate kill, and at flowering. And this is year 1, year 2, year 3. And this is our seeding rates of 0 all the way up to about 50 kilograms per hectare of hairy vetch. And what we wanted to see is what is the optimal seeding rates. A lot of this has not been done. We've been using cover crops for so long, but with the age of mineral fertilizers and herbicides, 50, 60 years ago, we knew this, but we don't know this information as much now. You'd be surprised how little seeding rate studies have been done, and more importantly, how much have them been done across broad regions to know how this varies. And so you can see here, just in my region, that obviously planting date a kill date had a big effect. If you killed it early versus if you killed it early, intermediate, or late, you can see the big differences in the amount of biomass that you can achieve with a cover crop like hairy vetch. And there's always going to be some differences between years because of climate, but generally you can see the potential holds. Now let's just look at the early and the late kill, but let's look at planting dates. So now this is when we planted it in the fall. So now you can see that the solid line is optimal. That's an early planting, and this is if you have to plant it late. Most farmers have to plant it later than they want. And so this shows you the potential difference between an early and a late planting on a cover crop like this, particularly in a colder climate. I guess I'm a warmer climate than some of these, but I'm still a little colder than most of you. But you can see the effect of the planting date. It makes a big difference. If you can get it in early, you make a big difference in the amount of biomass you can achieve. And seeding rate doesn't play an important role in my region of getting there. As you can see in my region, we can get a lot of biomass at a very low seeding rate. Where this line flattens out, where this line goes up and then flattens out right here, there's no value in adding any more seed after that point. It makes no difference. So we used to recommend to our farmers all the time 20, 30 kilos per hectare of seed. Now we're seeing we can get there with 5 to 10, which if you're a farmer, legumes are very expensive seed. If you can reduce your seed, you can reduce your cost and get more benefit. Now let's look at how this plays out from a colder to warmer climates. And you can see big differences, right away. That it doesn't matter how high you seed your cover crop over here, right? The seeding rates here, you can see the high seeding rate, your biomass potential is never as good as these warmer climates. And it takes higher seeding rates to get to your maximum potential. So you have to put higher seeding rates, and you have to put higher seeding rates, and you never get the same amount of biomass. So your dollars per pound of nitrogen is always higher. So if you're in a northern climate, no matter what you do, your dollars per pound of nitrogen that you get from your cover crop is going to be costing you much more than what you get when a warmer climate. So you get more money, you get more value in a southern climate, and that's important. This isn't going to work everywhere. Where you live affects how well this is going to work for you and fit into your crop rotation. Now, before I showed this schematic and I said, okay, there's a perfect cover crop out there and it does everything you want. But we know that that's not true. We know that these lines don't go all the way to the end, that if you're a grass, you don't fix nitrogen so that you don't get that benefit, whereas if you're a legume, you do. So we can target different objectives with cover crops. A grass is really good at suppressing weeds, whereas a legume is good at fixing nitrogen. So if we're trying to get multiple benefits, combining species has a lot of value. Combining species has a lot of challenges, too. So let's look at some of those challenges. So as I mentioned, grasses are good for weed suppression. Legumes are really good for providing nitrogen. Combining them, you can get multiple objectives. But how you do that depends on what your needs are and certainly your climate and your soil. So here's a figure. It's a complex figure, but we're going to walk through it together. This is hairy vetch to cereal rye seeding rates, and this is the ratio. So if it says 0, that means no hairy vetch. If it says 0.2, that means 20%, 40%, 60%, all the way up to 100% hairy vetch, versus no rye, all the way up to 100% rye. And this is the total biomass. So we're looking at experiment where I changed all these seeding rate proportions to show you how they behave together and what the total biomass can be. And these are big cover crops. This is not killing it early. This is when we kill it late, when they reach flowering, and this is a pretty fertile field, so we produce a lot of biomass. Now, this is a lot of figures. I don't want you to look at all these different figures. What I want you to look at is how different these figures look. This is 6 site years of data. I did the same exact genetics of cover crops, the same exact seeding rates on fields that are not that far apart climatically. But they behave very differently. In this year, you can see that no matter what you did, when you increased your hairy vetch seeding rate, it never produced a lot of biomass, whereas the rye, even at 20% seeding rate, it produced almost the maximum amount of biomass. 20% seeding rate of rye produced the maximum amount of biomass. Whereas in this year, you can see that the vetch produced a lot of biomass quickly, and the rye was very slow. So the point here is that cover crops are not a mineral fertilizer. They're not a herbicide. You can't drive across the field 9 mile, I can't do the conversion. You can't drive across the field very fast and apply a perfect rate every time. They respond to their environment. This field has a long history of manure, and so it's got very high nitrogen in the soil. And so when you have a high nitrogen environment, your grasses are going to dominate. Grasses are going to do really well if the soil is fertile. Legumes, who needs them if you have a lot of nutrients in the soil, right? They don't do as well, so they're not as competitive. But if you have a low fertility environment, the legumes are going to be very very competitive. So what this means is you have to know your fields. Cover crops are not a prescription. You can't tell a farmer what to do and say, this is how you do it. Go have fun. I look forward to the same results every time. It doesn't work like that. Farmers have to know their fields. They have to know how they differ in fertility, how they differ in drainage, how they differ in climate, because all of that affects how the cover crop performs, and this gets even more complicated when you're trying to work with mixtures. Trying to get the same results is very hard. So you might need to increase your seeding rates on a grass if you're in a low fertility site just to get it more competitive. Vice versa, if you're in a high fertility site, you want to lower your grass seeding rates a lot just to get the legume to be able to get going. All right, now we're still looking at these same ratios, right? We still have these same ratios of this is no vetch and this is 100% vetch. So this is pure vetch, pure vetch, pure vetch, pure vetch, and this is pure rye over here on the ends, right? And what you're looking at is how much nitrogen is in the biomass. And you can see that the vetch is the red line. The vetch increases in nitrogen with biomass. Makes sense. The more biomass you have, the more nitrogen. The black line, you see it doesn't do much. That's because the nitrogen content from cereals don't change much, regardless of the seeding rates and the biomass. And then you see, I'm sorry, I was mistaken. The blue is the vetch, and the red is the total. So the blue is your vetch increasing nitrogen with increasing seeding rate and biomass. And there's a dashed line in there. I'm not going to explain a lot about that, but that's how much biological nitrogen fixation you get. So that's just showing that the more grass you have, the less nitrogen fixation you get compared to how much is taken from the soil. But what I want you to look at here is that you can see that there's no biomass here. If you remember, this is our more fertile site where we saw the vetch was not very competitive, and this is our less fertile site. When we have more fertility, even though we seeded the hairy vetch at a high seeding rate, it never produced that much biomass. So that's why we have these big areas where the biomass didn't change, because this is not my seeding rate. This is actual biomass in the field now. So this is how much we achieved. So the point is some years you can't get that biomass because of the site conditions. But what I think is really interesting here is that whenever you achieve about 50% of your cover crop is a legume, so 50% of the mixture is a legume, you get the same amount of nitrogen content. See, this is the nitrogen content of the pure legume. This is the nitrogen content of the total biomass. And the total biomass, you can see it's about the same. And so you have the total amount of nitrogen in a monoculture of vetch, and the total amount of nitrogen in the mixture when it's 50-50 is the same. And we're going to see that this was my study that I did for many years at Maryland. Now I looked at data all across the U.S., and these are experiments all across the U.S., and we did a meta-analysis. And what we see here is this is the mixture in relationship to vetch and the mixture in relationship to rye. So if you're above zero, that means the mixture did better than vetch. If you're above zero here, that means the mixture did better than rye. And you could see that the biomass was always greater when you had mixture, and that the biomass was always greater than pure vetch, and it was often greater than pure rye when you grew them in mixture. When they were looking at nitrogen content, often you saw no differences between the mixture and the pure vetch, but obviously you always have more nitrogen, right? You always have more nitrogen when you have a mixture versus pure rye. So this just shows these results are consistent at many locations. And so this is just reiterating that same point. It's the same that when we have mixtures, we can often achieve more biomass than the monocultures, and that when the biomass of the mixture is 50% legume, we can get the same amount of total nitrogen. And we saw that this behaved this way across many different climates and different planting and termination dates. So we've talked a lot about the quality of the cover crop, how much nitrogen and carbon is in there. Let's look at now how these cover crops decompose. So this is cereal rye, and these are 2 years, year 1, year 2, and this is cumulative degree days. Degree days is just heat units. It's how hot it's getting, and so as you go through the summer, it's getting hotter. So just think of that as time, just increasing days. And so what you're seeing here is look at the small rye. This is small rye, only about 1,000 kilograms per hectare at the tillering growth stage, and that decomposes very fast. If you remember, we showed that a small cereal rye cover crop at the tillering stage has a carbon to nitrogen ratio of 18:1. So it's releasing nitrogen and decomposing very fast. This is 4 to 8 weeks before we're planting our cash crop. So if you worked hard to get that nitrogen by planting that cover crop and keeping it from leaching from the rains in the winter, but then you kill it early and it's a high quality material, you can lose that nitrogen still. It goes down into the profile. If you have a heavy, wet spring, you can lose that nitrogen you collected. So this is why there's a lot of advantage to letting the cover crops grow bigger. If the cover crops grow bigger, now the quality is not the same. It's a much higher carbon to nitrogen ratio, which means it's a low quality. And that decomposes slowly. Not only does it decompose slower, but less of it decomposes. Because look, this is not total biomass, this is the proportion. Right? So since it's a proportion, what I'm saying is that it decomposes slower and less of it decomposes. And you can see that this was consistent between years. You can have differences between climate that affect some of that rate, but generally they behave the same. Okay, now don't get angry with me. I have lots of farmers in the U.S. who still till, so this is for a tillage system, but I want to show you the difference between till and no-till. So now look, this is pure hairy vetch, this is a 50-50 mixture, and this is pure rye, and this is how much is decomposing and releasing nitrogen during the growing season. And I put these pictures in there to show you the growth stages that the corn will be at when it's decomposing. And what you can see is that when you till and incorporate that legume, it's exploding into the soil. It's exploding so fast that within 10 to 12 days, almost all of the nitrogen in that biomass is released into the soil. I'm not saying it's gone, but it's released into the soil and is becoming soluble. Versus, and that happens very fast. Now you can see the mixtures is a little slower, but the mixtures also produce less overall nitrogen. So this is how much nitrogen is available during the growing season, and you can see there's big differences between the mixture and the pure vetch. But let's remember what I was just talking about earlier. These two cover crops here, this pure vetch and this pure mixture, had the same amount of total nitrogen. The mixture and the pure had the same total nitrogen. But their release rate, how much was plant available, how much was available to the corn crop, is quite different because the quality was quite different. The vetch alone has a very low CN ratio, whereas in mixture, it has a much higher CN ratio, and so it decomposes slower and doesn't release as much. And rye, it really does not release much nitrogen. In fact, when rye is decomposing, sometimes fungi are actually using the biomass in the cover crop to move nitrogen back into the cover crop a little bit. So it's not going very far. Now let's look at what we're more excited about, no-till. And in no-till application, you can see now that you get the same amount of nitrogen that is released from the different cover crops, but you can see it's much slower. Now, this is a much slower rate of nitrogen release in no-till. And that's because the soil, when you till, the soil is very stable. Soil moisture, soil temperature, it doesn't change very much. On the surface of the ground, where our cover crop mulch is, in a no-till, it's not a very happy place to live. It gets really wet, it gets really dry, it gets really hot, it gets very cold, and that happens every day, twice a day, going up and down. So bacteria and fungi don't like that very much, and so that's why decomposition is slower and more synchronized with crop uptake. Now let's look what's going on in the soil. These are two different years, so year 1 and year 2, and this is soil inorganic nitrogen in the profile of corn, maize, at 30 centimeters deep, and right here, this 76 centimeters, this is a corn row to corn row. So you're looking at, this is a corn plant, and here's a corn plant, and we're looking at what the nutrients look like between them in the soil profile, 30 centimeters deep. And we're looking at that for pure rye, mixture, and vetch. Don't worry about these numbers. These numbers are just concentrations. So they're not put to total content. The total nitrogen is different than what the concentrations are. All I want you to do is see how different those colors look, how much nitrogen you get right away from a pure legume versus the mixture versus the grass. And then this, I forgot, I'm so sorry, I should have put this in here, I'm sorry. This is at planting. This is at planting. This is at V7. So this is V7 when the corn is about this high. And then this is when the corn is tasseling, when the corn is really tall. So this is just showing you time. So the top box is at planting, and then each box down goes later in the season. And it's just showing you that as you get in towards V7, now you see more nitrogen throughout this profile. Here you don't. And look at how interesting this is. See how there's not much nitrogen here? But those all have roots in them. All the roots go down into the soil well below 30 centimeters. So it's important to realize that the quality of the root is not the same as the quality of the shoot. The shoots have more nitrogen in them. So even though we kill this cover crop and there's nitrogen in the roots are decomposing much slower, and they're not putting as much nitrogen in that profile right away. Okay, now I want to shift gears and talk about some on-farm research work that we're doing. And this kind of puts together a lot of what I've been talking about and connects, I think, to the kinds of things that APRICID did. I've been touring with APRICID the last couple of days. It's been really wonderful to meet a lot of the team and the farmers that they work with, and they're doing a lot of really good work. And this just shows some of the kinds of work that we're doing on farm with growers in our region. So while there's a lot of interest in soil health and building soils, in the U.S. at least, farmers, the farmers who like to experiment are experimenting to try to get to soil health, but a lot of our farmers are more concerned about jumping into cover crops because of the economics, and there is a slow learning curve, and it takes time to get some of the benefits. So we're trying to focus on short-term benefits, just looking right in the immediate time, how does the presence and absence of the cover crop affect water and nitrogen, and then be able to make decision support tools through web-based apps, internet apps for growers. And so we've got funding to do this on farms throughout the East Coast here. We did this for 45 farms for the last couple of years, and we just got more funding now to do 75 farms, and we'll be doing this for another 5 years. And what we do is we go out on the farms and we measure the cover crops, we measure the soil, and we look at the water. So this is a water sensor system that we put in, and this allows us to look at the water at the surface, water about 30 centimeters, 60 centimeters, and down to a meter deep. And so we can look at the infiltration rates of the water and the total water over the growing season. And while we're going to do this for many crops, we're starting in just no-till corn. So all of this data is no-till corn. And then we're making apps for our farmers, not just for decision tools, but while we're doing the work. So while we're putting the sensors in the field, a farmer can go on the internet, they can go into our tool, they can type their name, and they can see their data in real time. So if they walk out into the field and they see strips of cover crop and strips of no cover crop, they can say, oh, that looks like the bare plots, the no cover crop, it has stress of water, and that one in the cover crop doesn't. Is that real? Am I just seeing things? And then they take out their phone, they look, and they say, oh, that's real. And so this is part of us learning with the farmer and gaining insights together. And so this is what some of our fields look like, of the cover crop and no cover crop. And this is what we do. We come in the fall before the cover crops are planted, and we take deep soil samples to a meter deep in the cover crop and no cover crop. And then we come in right before killing the cover crop, and we take those soils again so that we can look at how the nitrogen is changing over the winter. And then we come in and we harvest that biomass, and we put them in these mesh bags. You can see here we use these little clippers. We put them in the bags, we weigh them, then we put them in the mesh bags at the exact same amounts, and then we take one sample with us and take it to the lab to analyze it, and then we come back in 2 weeks, another 2 weeks, then 3 weeks, then 4 weeks, and then 5 weeks. And we take another bag. And this allows us to monitor the decomposition of the cover crop and how much nitrogen it's releasing. This is how I did all that other work I showed you earlier. And then this is what the water sensor systems look like. And then when the maize gets tall, we have to put the solar panel on a stick so that we can get sunlight and keep it going and keep getting the data. So now we're looking at how much cover crop biomass we're getting from the different farms. And I should have said earlier, we don't control anything the farmers do. We don't tell them what to do about their seeding rates, their planting dates, their termination dates, their fertilizer, their herbicides, nothing. They do what they're going to do. The only thing we ask is that they either don't plant a big strip of cover crop or that they go in the fall and they spray out a big strip with a herbicide so that we have a cover crop, no cover crop. This is what the biomass looks like on different farms. This just shows you that we had many farms, and most of the farms had 2,500 kilos, whereas only a few farms had these really huge cover crops. And the ones with the really huge cover crops are usually dairy farms or very close to chicken operations where we have so much chicken manure they have to go on the fields and the fertility is very high. Where we live, there's a lot of chickens. It's a lot of chicken operations, so we do a lot of work with chicken manure. So now this is what that looks like across the regions. As I said, I only have 1 year of data. I've only got this from my student 2-3 days ago, so this is the first I'm looking at it really. So more coming, but this is only 1 year's data. And as I said, now that we got more money, we're going to also try to do these other regions that we're missing and go further south. But you can see that where I live, this is where I live, you can see that we don't have as much biomass because it's colder. Whereas if you go down further south, for us, it's south, for you, it's north, you see more biomass in some of the farms, and that's not just because they killed it later, it's also because it's warmer. Right? And now let's look at some of the yields. Generally, now what you're looking at here is you see the zero. The zero is, this is the effects of the service crop on maize yields, and the zero means there was no effect. If there is a +1, that means that the cover crop improved yields. If it was a -1, the cover crop reduced yields. And what you can see is that generally there was not a big effect. Now, you can see that some places there were, and I think that's where we had a lot more biomass. If you remember the previous slide, the places that are brighter red had more biomass than the sites that are either yellow or turning blue. I guess that's maybe blue or purple. I don't know. So this is now just breaking it up by states. So this is where I live in Maryland, and these are going further south. So this climate is more like where we are here now or in Rosario, and you can see that there was some effect, that there was some lower yields across some of the farms, but it wasn't very significant. And that difference in some places were bigger than others, but it wasn't huge. And so why do we think that we may have seen that? We have a couple of ideas. I don't think that it's allelopathy. A lot of people think that allelopathy can play a very important role in affecting weeds and maybe even impacting our cash crop. But it turns out that allelopathy doesn't have a very big effect, and it really doesn't have a very big effect on our large seeded cash crops. I know this is a terrible figure. My colleague sent me this figure, and you can't see any of the numbers and what those words mean. But what this is basically a chemist analyzed all of these different chemicals that are in the soil and looked at how much concentration there is. And don't worry about the numbers. The important thing here is this is just the roots, and this is just the shoots. So I went into a bare ground soil where there was no cover crop, and I picked up all these shoots, and I put it where there was no cover crop, and then we went to where the shoots and the roots were, and we removed all of the above ground shoots when we killed everything, so we could just look at the effects of the shoots and the effects of the roots. And the important point here is that, first off, where we did see a lot of allelochemicals, they're gone within 2 weeks. This is week 2. So this is 14 days right here. 14 days. Within 14 days, there's very little allelochemicals in the soil. And you can see from the shoots alone, there's almost none. Very little. So most of our allelochemicals come from the roots, and most of it is gone within 2 weeks. So we don't think these yield differences, because most of these farms waited at least a couple weeks, were allelochemicals. What I think it is it has a lot to do with nitrogen. In our region, There's a lot of concern with water quality because we have a lot of nutrients, excess nutrients often, that are going into our water supplies, our groundwater, and then causing problems and hurting fisheries and recreation and more. And so there's a lot of emphasis to address water quality. And so our farmers now, to increase their nutrient use efficiency, are splitting their nitrogen application. They don't put that much down at planting. They just put a little bit down at planting, and then they put most of it down at side dress. They put it down at V7, V5, when the corn is knee high. When the corn is knee high, that's when they put down 75 to 80% of the nitrogen. So when you're used to putting just a little bit of nitrogen down early and not a lot later, that works fine if you don't have a cover crop. But if you're planting a big cereal rye cover crop, a cereal rye cover crop ties up all that nitrogen and it's not available early on. And so our farmers who've been spending a long time now splitting their applications, what we've been working with them on is to change that split. They're going to have to put more nitrogen at planting now if they're going to plant bigger rye cover crops. And so now on this on-farm experiment this year, we put out micro plots where we put down an additional 50 kilos of nitrogen at planting so that we can make sure, is this something else or is it nitrogen? 5 minutes. 5 minutes total. Yeah. So I only have 2 or 3 slides left. I just wanted to mention something about water. I was going to just show you that because we also collect a lot of information on water. And this is just a little snapshot. But this is all no-till ground. And what you're seeing here is the effects of a cover crop. This is a cover crop, and this is no cover crop. And this is rainfall intensity. So this is when you get 20 millimeters of rain in one event versus 40 milliliters of rain in one event. So this x-axis is increasing rainfall intensity. And what you see is that even in no-till ground, when you have a cover crop, all the water infiltrates and the soil stays wet, so you capture that water. And that's important in the summer when it's drought, is you keep that water. But what you can see is when you have no cover crop, even in no-till ground, without the cover crop, as the rainfall intensity increases, the soil starts to dry, which means less water is getting into the soil. And the main point here is that that no-till is not enough. That cover crops are an important part of what we are trying to do with no-till agriculture. And this just highlights that on a summer like this, you can see this is cover crop, this is no cover crop, and that is where we saw some benefit of water, the increased water availability to our crop. Now, sometimes when we do research, we're lucky. Sometimes we do research, we're unlucky, right? So where I live, we sometimes have 3 years are wet, 3 years are dry. So 5 years ago, if I started this experiment, we would have had perfect place to test this because it was wet. It was dry. So we could see the benefits of a cover crop in a dry year. The last 2 years in our region has been very wet. So that's partly why we didn't see much of a difference in these cover crop effects, because we had a lot of water and we had no water stress in our fields. And they asked me to just mention briefly also, this is my last slide, that we also now, to improve cover crops performance, we don't just focus on management, but we're also doing a lot to improve the genetics of our cover crops, because we think that cover crops have received very little improvement. When you want to make an improvement in maize, you need millions and millions and millions of dollars and many years to do that. Cover crops have received very little improvement, so we think we can, for less money and in shorter period of time, we think we can see bigger improvements in the genetics and help farmers achieve even more results with the cover crop. So just to review, there are short-term and long-term benefits. Some of these short-term benefits provide immediate dollars, But it's important to realize that if you're just planting a cereal rye cover crop and you have a small amount of biomass out there, many years you're not going to have a yield difference. Some years when you have drought, you could see yield differences. Over years, as you build your soil health, you'll start to see some more. So cover crops are a commitment. You have to be patient. A lot of the benefits come after 3, 5, 7 years. Thank you.
Bueno, hola, hola.
Oh.
Hola, hola, hola.
So we have many questions. First question, Steve, how do you see the use of cover crops for production of energy?
The question was, how do I see the use
Of cover crops to of energy, correct.
I keep hearing myself. Energy, meaning like biomass for biofuels. I don't do much in the biofuel realm, so I don't want to speculate and say much. Obviously, if we increase the performance of our cover crops and can reduce the costs, they can play a role in that. But I don't do much in biofuels, so I don't want to say much.
¿Han realizado algún estudio sobre
Have you done any study on the effects of root growth and the formation of organic carbon?
The question was, do I do much studies on root growth and the effects of carbon? Everybody always wants to know about the role of cover crops in sequestering carbon, and I couldn't agree more. That's a very important question. The problem is that carbon sequestration takes a lot of time. You don't see changes in 2 or 3 years. You see changes more in 5 to 8 years. And that really depends if you're in a cold, heavy soil or if you're in a warm, coarse soil. Or, so if you have sandy or clay, if you're warm or you're cold, those play huge differences in your ability to sequester carbon. And so we need long-term cropping systems experiments to do that. And there are very few long-term cropping systems experiments out there. And the ones that do have cover crops in them, many of them have multiple differences in the experiment. And so it's hard to know exactly what is the effect. There are some now, at least I know in the US, that are achieving 3 to 5 years where they controlled everything. And there are some that have been a little bit longer and have seen differences in carbon. But this is slow, and we have very little information across a lot of different sites. So it's hard to say anything specific. We do know that roots are more important than shoots for carbon sequestration. That we get much more carbon storage in the soil from roots than we do shoots. And what some scientists are looking at is how decomposition affects the types of products released from microbes. And interestingly, we're seeing that it's important to have legumes in with the grass, that grasses actually don't sequester carbon as fast alone as potentially with grasses and legumes together. And this is an area of recent development, but they think these scientists think is that the decomposition is much more intense and aggressive when the grasses are with legumes, and so you can get more sequestration. I know that did not answer the question, but at least I gave some information about that.
¿Tienen información?
Do you have information on the use of different kinds of cover crops to improve high pH soils, soils that have structure problems?
Do it again. effects of cover crops on high pH. The question was, have I looked at the effects of cover crops on high pH? This is a tough crowd. No, I have not, so I can't say much about that. Sorry.
Could you measure other benefits of cover crops? For example, the decrease of weeds, or in order not only to think about the next crop or the yields of the next crops or the following crops?
The question was, how does cover crops affect weeds and subsequent crops? And we do a lot of work on that. Some of my formal training is in soil fertility, weeds, and I do a lot on water. So we do a lot in weeds, and generally we see a lot of benefit with cover crops on weeds. It depends on the species. We have a lot of success with cereal rye, especially for us. Cereal rye sticks around a lot longer. Legumes decompose very fast and can stimulate weeds to grow because weeds are stimulated by nitrogen, and so we have much better weed suppression with grasses. And we find that you start to see benefits when you get above 2,500 kilos per hectare. When you get lower than that, sometimes for us, at like 500 to 1,000, we stimulate weeds to grow because there's not enough biomass to suppress weeds, but there's enough biomass to create microsites for water. And so we see more moist conditions at low biomass, and sometimes we can stimulate weeds and give them more benefit. Whereas once we start to get above 2,500, we start to see effect. We really are trying to target 5,000 to 7,000 for really extensive weed control, but we also see a lot of really good interactions with herbicides where we even see synergistic effects that weeds that are harder to kill with just herbicides alone, that together with the cover crop, we get a very high degree of control. And this is important particularly for biennial species like Caniza, because when the cover crop is establishing in the fall, so does that weed, and it can outcompete that weed, and we see huge benefits for Caniza, which has resistance where we are, and so it can provide a lot of benefit. But there's a lot of research on cover crop effects of weeds. In the US, we are seeing massive adoption on cover crops for that purpose. All along the southern part of the US, where resistance is the biggest in the US, those were regions that we did not historically see a lot of cover crop adoption for cereals, and now all of a sudden you see farmers all over these regions starting to use cover crops to help with suppression. And what's really nice about cover crops, because even though they don't suppress all weeds, cereal cover crops particularly, but cover crops in general, are very good at suppressing small seeded summer annuals. And so what's one of our biggest problems is amaranthus. It's a small seeded summer annual, but that has a lot of resistance in it. And so the fact that cover crops are very good at suppressing small seeded summer annuals, there's a lot of advantage to having that in the system.
How can we know the amount of kilos of nitrogen that fixated the soil?
I wanted to just add one point to the previous that I forgot to mention. It is important to note that cover crops, mulches, have very little to no effect on perennials. If you've got a perennial weed in the system, they do not receive much suppression from a cover crop mulch, and so there's going to have to use other tactics. The last question was about how do you know how much nitrogen is in the cover crop and how much is going to be available during the growing season. So what I've showed you today was work that we are measuring this in the field. We're measuring it on farms, and we're developing models. What we're trying to do is develop models that will work across many different climates and soils. We're trying to make a tool that can work across our whole country, but give very site-specific recommendations based on your climate. And this is because we're using the quality and the quantity of the cover crop to make those decisions. So we think that in the near future, we will have tools that a farmer can just take a sample of their cover crop, send it off to the lab, get a quick analysis, and then they can submit that analysis into a web app, and the app is going to give them a release curve of nitrogen that's available. And that's a big part of where we're going, is to provide decision tools for growers. But generally, the way we think about how much is available is often we see about 50% of the total nitrogen in the cover crop is available to the cash crop in that growing season. So it's a crude number. If you're working with a pure legume, that number goes up. If you're working with a pure grass, that number goes down. But the mixtures are about in between. So that should give you a sense, 50% depending on a pure legume or grass.
Bueno, vamos con la última pregunta.
Last question, because we are running out of time. Have you considered the possibility of deciding the drying of the cover crops due to the consumption of water of the profile? Or do you consider just what other variables do you consider when you decide on the drying?
So I think the question is asking, how do we take into consideration the effects of cover crops on water or the drying of the soil? And I'm assuming the question relates to how that would affect the subsequent cash crop. What I hope I've impressed upon you today is that researchers doing a project at one site is interesting, but it has very little value broadly. If we're going to know anything about cover crops and be able to provide site-specific recommendations, we need to do these studies at lots of different climate and lots of different soils, or we won't know the answers to those questions. So in my region, we have really good spring moisture. So generally, even after we kill the cover crop, very quickly the soil profile recharges and we don't have limiting water, but we have very droughty summers. So our springs are wet, our summers are dry, so when we get into the summer, we get a lot of benefit. And for us, the farmers really like the cover crops because they actually like the drying. They like, when the soils are wet, it's hard to plant, and so they like the cover crops because it dries the ground faster, it gets them into the field earlier, and you have less compaction when you plant with the cover crop on the surface. So for farmers in my region, they like cover crops because it helps reduce water. But other regions are going to be different. And that's why this work has to go on at all of these different regions so we know how it has that effect. So that in certain places, you're going to have to act differently. You might have to wait 2, 3, 4 weeks, maybe longer before you plant into that cover crop to make sure that the rains recharge your soil. And so this is not just a researcher's objective. This has to be the farmer working with the researcher, because cover crops are not just a science or very precise, it's an art. And you need to be thinking all the time.
Bueno, estamos con el tiempo cumplido.
Okay, so we are out of time. I would like to thank Steve for his participation, and we are concluding this panel. Thank you very much.